All right, hello everyone. We are here to do the Identifying Carbohydrates Lab, and so again, I'm just going to model that for you, and we're going to go through it kind of step by step. And again, your job is to do the write-up according to the lab report format sheet that you can find on the module. There's a little button there for it, or you can also find it back in the Unit 1 notes and handouts. Our purpose of this lab is we are here to identify what these three unknown substances are. So we have substance A, B, C, and we're trying to figure out what they are in terms of a monosaccharide, a disaccharide, or a polysaccharide. So as you remember from the notes, monosaccharides, they will test positive in the presence of Benedict's solution, and polysaccharides will test positive in the presence of iodine solution. And we don't have a test for disaccharides, so we're going to use the process of elimination for that one. And so we're going to use our knowledge in those things to try to figure out which substance is which here. And so again, that's our purpose. Your hypothesis, what you're going to guess, is just tell me what you think each one is just by kind of looking at it. So substance A is kind of a cloudy looking mixture. B is clear. And C is also clear. So again, I know these are just pure guesses, but you're just going to guess which one you think is mono, di, and polysaccharide. So remember, hypothesis doesn't have to be right. So as long as you have a guess there for all three of them, then you'll get the points for that. And so safety, we are dealing with test tubes and test tube racks. If you were in class and you had to carry these around, you would have one hand on the bottom of the rack, one hand on top. That way nothing could accidentally slip out of the rack. If we carry it like this, they're kind of loose in there and shake around, and something could actually slip out. We don't want that to happen. So we want to make sure we're carrying these carefully if we're in class. The other thing is we are dealing with chemicals, so we are going to have our goggles on. So I'm going to go ahead and put mine on right now. Remember, safety goggles are a little bit more protective as opposed to just normal eyeglasses. Eyeglasses are just kind of shielding the very front of the face, but other stuff could still drip in. These are splash proof, so those are going to be my safety goggles for the day. So in terms of this, the first thing we're going to go ahead and do is we're going to get the Benedict's test started, and because that one takes a little bit of time. And so all I would do is I would take just a sample of each of these, and so I don't want to mix up my droppers, and so I would just put A in my first test tube, and I just need a little bit just to coat the bottom of the test tube. And then I'm going to put some B in my second test tube, and I'm going to put some C in my third test tube. And then I need to put my Benedict solution in there. My Benedict solution starts off this nice blue color, and if there's no monosaccharide in there, it will stay blue. If there is monosaccharide in there, it'll start to turn sort of a greenish color, and if I leave it in the heat long enough, it'll turn to like a rust color. But it says for 2 mLs to go in there. An eyedropper is about 1 ml, so I'm going to do 2 like full squirts in each test tube. And then the next thing is, in order for Benedict solution to react properly, I need a heat source. It has to be warm water. It can't be boiling. If it's boiling, it breaks down the molecules, like there might be a polysaccharide. It'll break them down into monosaccharides. So I don't want to get a false positive test. So I just want the water to be warm. I don't want it to be boiling or anything. And I've preheated some warm water right over here. So we're going to take these and we're going to keep them in order so I can keep track of which one is which. So this one over here is going to be A. And then my next one right next to it will be B. And my third one right next to that will be C. And I'm just going to let them sit there in that warm water while I do the iodine test, because that takes about 5 minutes or so. So we're going to go ahead and just let that sit there, and we'll do the iodine test next. Okay? So let me put this off to the side. For the iodine test, we're going to do the same thing. We're going to do a sample of each. So again, just 1 squirt of my A. Just a single squirt of B. And a single squirt of C. And for my iodine test, I only need a couple of drops of this. It starts off kind of an amber color. Let me see if I can show that to you. Amber is kind of almost kind of a orangey yellowish color, kind of in between the two. And I just need a couple of drops in this, and this happens immediately. So I put in 2 drops there, a couple drops here, and a couple drops here. And so you can see, if I kind of mix those up a little bit, you can see right there, hopefully, in the white, there we go. So my A turned black, but my other 2 stayed amber. And so I'm going to go ahead and put that on my data table. So again, I'm doing the iodine test. And so A was black, B was amber, and C was also amber. Okay? And so my iodine test is now done, and so I don't need to worry about that anymore. So now let's go ahead and look and see what we got in our test tubes over here. So I'm going to take these out in the same order that I put them in. And so because they're warm, I'm not going to touch them with my hands this time. I'm going to go ahead and grab them with this little tongs. And so that was A. And then this was B. And this was C. And so you can see from those results that A and B stayed blue, but C turned sort of a rust color. And so let me go ahead and get that on my data table. So for the Benedicts, A stayed blue, B stayed blue, and C turned rust. Okay? So now on your document, you're going to go ahead and fill in that data table. And you're going to use the same colors that I used here, and you're also going to fill in the third column. So based on those results, you should be able to tell me is each one a monosaccharide, a disaccharide, or a polysaccharide. And so that's what you'll get graded on in terms of filling in the rest of that data table. After that, you have some questions to answer and a conclusion to write as well. And so again, your conclusion should follow that CER format. Again, look at the lab report format sheet in order to help you with that. Don't forget, the last time a lot of people left off restating the purpose and the hypothesis. So don't forget, you start off your claim by restating your purpose and your hypothesis, and then you go on to explain your data and why your hypothesis was right or wrong. Okay, and so don't forget the why part as well. All right, and so that's it for the carbohydrate identification lab, and good luck on doing your write-up.